Yeah, let's get started. So Dr. Maria Geffen joins us today from the University of Pennsylvania, where she's a professor of otorhinolaryngology. I practiced that a few times, neuroscience and neurology. Before Penn, she received her academic training at Princeton, Harvard, MIT, and Rockefeller University. Her research broadly encompasses the study of neuronal circuits for auditory perception and learning. And to do so, she uses a number of methods from the systems neuroscience toolbox, from two-photon calcium imaging to more behavioral and computational approaches. Some of her recent results include identifying a novel cell type that supports hearing of unexpected sounds, establishing the connection between emotional learning and sound perceptual acuity, and elucidating the neuronal code for representation of communication signals in the brain. Her findings have been published in a number of well-established journals, such as Nature Neuroscience and PLOS Bio, and have been recognized with a number of prestigious awards, including the Burroughs Wellcome Career Award as the Scientific Interface, the Klingenstein Foundation Award in Neurosciences, the Auditory Neurophysiology Young Investigator Spotlight Award, and the Kaufman Foundation New Initiative Grant. All of that said, please join me in welcoming Dr. Geffen as she shares her insights on the role of cortical inhibitory neurons in auditory computation.
Thanks so much. What a great introduction. And it's really good to see everybody here, some familiar faces. I was trying to remember, I was at Georgia Tech before the pandemic at some point, but I don't remember the year because my memory has been blurred. Anyway, so I'm gonna tell you about some of our work on the role of cortical inhibitory neurons in auditory computations. So just to kind of shift you to the domain of auditory perception, I'm going to play you a piece of music. Hopefully it'll... Oh, it did not play. Okay. Well, you have to imagine this pianist playing a beautiful piece by Rachmaninoff. And as you're listening to this piano piece, your brain is actually performing a number of different computations. One of the most basics is sound frequency discrimination. So on a piano, most of you know that there are different notes and they correspond to different frequencies. So for example, on the right, you're going to have high frequency notes. On the left, you're going to have low frequency notes. And so a lot of what auditory computation entails is decomposing the signal into frequency delimited bands. So this would be kind of like picking out individual notes from the incoming sound stream and comparing those transformations to each other. In a way, if we think about the cochlear is kind of like an inverse piano. It takes in the incoming sound stream and is comparing it and is producing a frequency delimited representation of the sound signal. So that's computation number one. Another very important computation is identifying statistics in the sequences of sounds. So as you're listening to me talk, much of how you hear me really depends on kind of you picking up statistical regularities in what in the sound of my voice. If I started talking with a very low voice, you would have very strong brain activity signaling violation of statistical regularities. If I said something very unexpected, a similar process would be happening. So this detection of statistical regularities contributes to predictive coding in the brain, and that's a really fundamental aspect of auditory computation and auditory cognition. So what my lab tries to do is to understand and map the neuronal basis for auditory computation. And we're very interested in studying this at the level of neuronal cell types and neuronal ensembles. We focus on the auditory cortex, which is the part of the sensory cortex that deals with sounds. It's generally thought that the auditory cortex is really the central hub for the transformation of sound representations, where we move from representing the sound waveform to more object-based and behaviorally uh, relevant representations. And we're really interested, and it's comprised of a number of different cell types that I'll describe to you in a second. And our goal is to really bridge our understanding of how auditory computations are carried out in the brain, from understanding how single neurons work to a full brain representation. And we focus on the sort of intermediate level that I know many of the laboratories here also work at, which is the level of neuronal ensembles. This is a slice through the Bowers cortex, and you can appreciate that there is many different cells, and there is a little bit of structure to it, although there is also a huge mess of different cells. What we try to understand is to identify within this kind of large scale neuronal ensembles some microcircuits that would be carrying out some of these more canonical computations. And we believe that's important because that should, in principle, translate, not just be relevant for sound perception and auditory sensation, but should also translate to other forms of sensory perception and sensory learning and cognition. We have been always interested in interactions between the excitatory and inhibitory neuronal populations. So for those of you who have come more from an engineering background, actually a lot of how we think of the brain, or we think about computation, relies on something more akin to excitatory neurons. So those are neurons that transmit informations between different sensory regions, perform computations such as integration of incoming information, etc. However, in the mammalian brain, there is a really important role for inhibitory neurons. So inhibitory neurons, when they get the signal, they actually send the signal. When they get activated, they send the signal to suppress, which suppresses the downstream units. And computationally, that's very important. So inhibition plays a number of roles. If you take inhibition out of the brain kind of, you can get seizures, you can get all kinds of abnormalities in sensory in processing of information. And this is something that has been studied computationally, although a little less, not as, let's just say, it hasn't been as prominent until the last few years in trying to understand how excitatory and inhibitory neurons interact. But if we put together an excitatory unit together with an inhibitory unit in a mutually connected circuit, that very simple system can actually produce a number of very interesting behaviors. In the brain, if those inhibitory neurons are substituted with different types of inhibitory neurons, inhibitory units, so with different connection properties, different types of rules for plasticity of the synapses, you can get an even more sophisticated network. And something that we have been pushing in our work is to try to integrate all of the results from our experiments and from experiments from other laboratories to understand the complexity of excitatory and inhibitory interactions in the auditory system, and try to understand what kinds of microcircuits they actually build, whether we can understand that at the a level of more mechanistic models, circuit level models. So I'm going to tell you I'm going to go a little historically through our work and tell you about what we have discovered and are continuing to discover about the function of different inhibitory neurons in auditory computation. I'm going to tell you that, not surprisingly, I wouldn't be starting with frequency discrimination if we didn't find that cortical inhibitory neurons actually control this very fundamental auditory computation, frequency discrimination acuity. Different inhibitory neuronal subtypes actually differentially control various aspects of auditory adaptation and auditory um, uh, statistical detection and novelty responses. I'm going to show you our work on the circuit models for excitatory inhibitory interactions. And I'll show you a recent study on the differential role of inhibitory neurons in controlling population dynamics more generally in the cortex. The very much simplified circuit motif that we have been focusing on is kind of depicted in this diagram. And I'm going to take you through. I apologize for all these abbreviations, but once you get used to it, and many of you have encountered them in recent literature, it's actually not so bad, right? So we consider excitatory neurons. So this is kind of a diagram of the biker circuit we're studying in the auditory cortex, where the excitatory neurons receive information and also send out information to other brain regions. And VIP, somatostatin, and PV neurons are different types of inhibitory neurons. And those are the three most prominent groups of neurons in the auditory cortex. The parvalbumin neurons suppress the excitatory neurons at their cell bodies, whereas the somatostatin neurons suppress the parvalbumin neurons and also inhibit the excitatory neurons, but targeting predominantly the distal dendrites. The VIP neurons are another type of inhibitory neurons, which actually inhibit the inhibitory neurons. And this has been shown through a very large number of papers. This kind of basic circuit has been mapped out across different cortical regions. So it's not specific to the auditory cortex. Depending on who you ask, these are like three major classes, but if you look for subtypes of inhibitory neurons, people have mapped up to hundreds of different inhibitory neuron subclasses in the human brain. It's smaller in the mouse brain, but there is still subdivisions of these classes, and there are other types of inhibitory neurons. But in our work, we have kind of focused on these three classes of neurons, and this has already kept us busy for about a decade. So our first experiment was to try to see how inhibitory neurons control frequency discrimination. And so the task of frequency discrimination, you can kind of think of it as if you're playing the piano and you play two distant notes, you can tell the frequency differences between those notes pretty well. And in fact, most of us can tell the differences between two adjacent notes pretty well too. But imagine that you tune the piano so that the difference between two adjacent notes becomes smaller and smaller until it virtually disappears. So there is some frequency difference, some frequency limit for all of us, and it varies across individuals, where we can no longer tell the frequencies apart, or at least we can't tell them apart as well as we could. And this is something that we can measure in mice using there are a number of different methods, but one of them is called pre-pulse inhibition, a modified version of pre-pulse inhibition of the acoustic startle response, where we play a tone at one frequency to the mouse, we switch to a different tone, and then we play loud noise. And we measure how much the mouse startles by that loud noise. And if it can detect the frequency difference, it startles less by this pre-tone than if they can't. So using that, we can basically map what we call a psychometric function, where we can have some measure of behavioral response as a function of the frequency difference. Now, what we were interested in understanding is whether inhibition in the cortex actually controls frequency discrimination. And so in order to do that, we have to have some sort of model of where this frequency discrimination or frequency sensitivity comes from. And at some level, the like the threshold for frequency discrimination should have some relationship to frequency tuning of neurons in the cortex. So neurons in the auditory cortex respond to frequency, to tones of different frequencies with roughly bell-shaped curve. There is some frequency they preferred, and there is some range of frequencies to which they're still responsive. Interestingly enough, because neurons in the cortex receive inputs from both excitatory and inhibitory neurons that themselves are tuned, really this width of tuning is controlled by the relative ratio of excitatory and inhibitory inputs that are coming in. So we wanted to do an experiment where we change this inhibitory excitatory ratio in the cortex. And we could do that using optogenetics. We would either activate or suppress the parvalbumin inhibitory neurons. What we expected that to do was that if we increased inhibition, we expect the frequency tuning curves to become more narrow and to increase the signal to noise ratio in cortical responses. And then we expected the behavior, the frequency discrimination behavior, to improve. So we expected the mice to be more sensitive to smaller frequency differences when inhibition was upregulated than when inhibition was decreased. So we could, in a separate set of mice, we could decrease inhibitory neuronal activity using archirhodopsin, which is a suppressive opsin. And we expected that the tuning curves would become broader. There would be lower signal to noise in the cortical responses, and frequency discrimination threshold would increase. So mice would get worse at frequency discrimination. And that's exactly what we saw. So on trials, when inhibitory neurons were upregulated, mice actually became better. They improved their ability to tell apart tones of different frequencies. On trials, when inhibitory neurons were suppressed, mice became worse at telling apart different frequencies. This was a really interesting finding. It's very cool that it was bidirectional because we could really establish the causality between cortical activity and behavioral performance. And essentially, we also showed that if we did a similar manipulation on just excitatory neurons, we actually did not see any changes behaviorally, even though the cortical activity overall was upregulated. So this showed us that cortical inhibitory neurons control the very fundamental auditory computation of frequency discrimination. But why are there different types of inhibitory neurons? Our next question was, well, what is it that different types of inhibitory neurons could be doing differentially in controlling responses to sounds? And here we focused on somatostatin neurons, and I'll later show you also the VIP neurons. We turned to this question of how does the brain extract information about sequences of sounds, and specifically how the brain extracts information about statistical regularities. At the very basic level, this is studied using the so-called oddball tasks. So in an oddball task, we play two tones. One of those tones in a randomized sequence, but one of those tones is presented very rarely, whereas the other tone is presented very frequently. It sounds something like this. Tune, 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 tune. So as you were listening to me, play this stimulus to you, some of you, I bet, were getting really, really bored by hearing the tune, tune, tune being repeated. And when I said tung, maybe And I bet I actually know that in all of your brains, the activity in the auditory cortex shot way up. So hearing and the brain, the auditory system really adapts very quickly to the repeated tones, so to something that's statistically super predictable. And there is also an enhanced response to an unexpected tone. So if we take a random neuron in the auditory cortex and consider the responses to which the neuron usually, to two tones, it's equally responsive. When we present one tone more rarely than the other, the response to that tone alone shoots way up. And as a control, we flip that relationship. So now we play the high frequency tone more often and the low frequency tone less often, and that the response to the low frequency tone becomes stronger. So this is called stimulus-specific adaptation. And in the auditory cortex, 95% of the neurons exhibit stimulus-specific adaptation. So it's a very fundamental feature of cortical neuronal responses to sounds. In a way, it's also a generalized phenomenon that has been found in the visual cortex and somewhat modified in the somatosensory cortex. And it's thought to be a form of notched adaptation. So in notched adaptation, if a priori we have some tuning curve of a neuron, but now one subset of the stimuli is overrepresented, what we get instead is a notch in that tuning curve. And if it's a low frequency tone, we will get that's underrepresented, we will get or if it's a high frequency tone that's overrepresented, we'll get a notch at high frequencies and vice versa for low frequency. Even though 95% of neurons do that, the mechanism for that has actually not been identified. And there were multiple theories for how this could come about. One of the theories is that it could be adaptation that's inherited in a bottom-up fashion. If we consider a cortical neuron receiving inputs from thalamic units that are more narrowly tuned, one of those units might simply might adapt to the overrepresented tone. And then if you're integrating the information across all of these inputs, you're going to have a notch for that particular frequency, that's been adapted. An alternative theory was that there is a tuned intracortical inhibition that contributes to reduced response to a specific frequency, to the overrepresented signal. And it's really this mechanism that we wanted to focus. So in order to investigate this, we suppressed either parvalbumin. So we presented this oddball stimuli to mice and recorded cortical activity. But on a subset of trials, we suppressed either somatostatin or parvalbumin neurons. Our expectation was that if indeed these neurons are responsible for this adaptation, we expected on trials when those neurons were inactivated, the adaptation to recover. So the neuron would respond just as strongly to the tone that was overrepresented as when it was underrepresented, and vice versa. And that's indeed what we saw. So here is a neuron that shows almost perfect stimulus-specific adaptation at baseline. But when we suppress inhibitory neurons, that adaptation actually recovers. We found that, in fact, inactivating either parvalbumin or somatostatin neurons drove a reduction in the stimulus-specific adaptation index, which is basically the ratio of the responses. But what we found is that the time course of the effect of this inactivation was very different. So in order to investigate that time course, we looked at the trials in our very long stimulus sequence. So here in gray are all the tones that are the repeated tones, and in red are all instances of unexpected tones, we picked out 5 trials just after the unexpected tone. During those 5 trials, actually, normally, neurons would exhibit a secondary form of adaptation. So presenting an unexpected stimulus actually relieved some of the adaptation to the repeated tone. And when we separately inactivated these neurons over the course of this adaptation, we found that inactivating somatostatin neurons recovered the responses proportionally to the level of adaptation of the neuron. So the more the neuron was adapted, the greater the effect of inactivating the somatostatin neurons. In response to the unexpected tone, there was no effect of inactivating the somatostatin neurons. So it seemed that the somatostatin neurons were in fact providing inhibition at the same as that contributed to this adaptation level of the neuron. So it was in a stimulus selective fashion. By contrast, inactivating the parvalbumin neurons simply recovered the neuronal responses regardless of their level of adaptation. And again, this is something that's consistent with our result for frequency discrimination, where when we inactivated the parvalbumin neurons, the responses in the cortex became less selective and less precise. Yeah.
Activate these populations, is there a change in the spontaneous firing rate?
Mm-hmm. Yeah, so this is all the difference relative to the spontaneous firing rate. Yeah.
What is the change?
Well, it goes up, so spontaneous, but it probably goes up. Well, so I will show later because we looked more at the level of kind of cortical activity, like a network level. So there's differences because they do different things. Yeah, it doesn't, but with PVs, you really just have kind of like removal of inhibition and just the general noise goes up. And with somatostatin, the effects are much more subtle than that. Yeah. But yeah, thanks. I love it if people ask me questions too. So please don't be shy. If you don't understand something or want more information, please feel free to interrupt. Okay, so this study showed us that somatostatin neurons were actually providing selective inhibition that contributed to stimulus-specific adaptation. More recently, we became interested in more complex aspects of statistical regularity detection and novelty responses in the auditory cortex. And in particular, we were interested in sound sequences. So here, if I present to you A, B, C, D, A, B, C, D, A, B, C, D in a repeated fashion, you actually are adapting not just to the frequency content of A, B, C, D, but also to the regularity. Versus if we compare it to randomly permuted sequence of the same sounds. In humans, it has been shown that if we present a regular sequence and then a novel stimulus, the response to that novel stimulus is going to be stronger if the sequence that was shown before that is repeated exactly in a regular fashion versus if it's randomized, like the way I have it here in the random context. You guys can tell the difference, right? So in both cases, I'm repeating the same sound units, but in one case, I'm keeping the sequence the same for the repeats, and in the other case, I keep the sequence different. From stimulus-specific adaptation, when I play a new sound, which I call the deviant, the X, Y, Z, W sound, we expect a novelty response just because it's a mismatch in the type of sound it is. But we also get a secondary novelty component because it not only breaks the kind of the ensemble of sounds that we're adapted to, but also the sound sequence. And so studies from Maria Chait lab have shown that, in fact, you can map that to activity in the cortex. So we tried to adapt the stimuli to the two mice, and I'll play you what we came up with to try to track the neural correlates for this increased activity. So. Can you guys hear that? So these are called dynamic ripples, and they're frequency ripples. So these are stimuli which span a broad range of frequencies, but they also have these changes in frequency kind of modulation. They activate, the reason why we pick them is that they activate very large populations of neurons. Almost all neurons are responsive to some subset of these stimuli. And if I play now the random context stimulus, okay. So it's the same deviant stimulus, it's the same unexpected sequence, but whether it's embedded in a regular or random context differs. So, and for those of you who play video games, you're probably used to hearing these sorts of things. I know my kids, there are sounds that come out of the room that are very similar. Mice actually, can learn to detect these sounds, although that is an ongoing sound in the lab. But we have measured just at the level of neuronal responses. What we wanted to understand is whether these novel stimuli exhibit differential responses under regular and random context. Oops. And whether they're controlled by inhibitory neurons. And as control, we just played these sounds separately, so not as part of a sequence. So, in order to capture the activity of a large population of neurons, we use two-photo imaging of calcium activity. So this is a technique that allows us to actually visualize the activity of hundreds of neurons. And we focused on the two primary auditory fields, A1 and AAF. And at the same time, we used optogenetic manipulation to suppress the activity of either PV, SST, or VIP neurons during the novel stimulus. And our goal was to try to see whether there's going to be differential effect of suppressing the activity of these different types of inhibitory neurons. So first, if we line up the responses to the novel sequence in different contexts, we can see that the response is actually stronger in the regular than in the random context. So the novelty response at the level of individual neurons in the auditory cortex reflects what we see at this gross population level in humans. Indeed, there is a separate novelty component that is probably due to the regularity in the sound sequence. And we can see that statistically over the population of neurons that we're examining. So now what happens when we suppress inhibitory neurons? Well, we found a very interesting effect for suppressing the VIP neurons. When we suppress the VIP neurons, the difference, so this right now you see the difference without neuronal suppression, and you can see that there is a strong novelty response in the regular context in cyan, and a less strong novelty response in the random context in purple. When we suppress VIP neurons, we still see the novelty response. However, now it's the same under the regular and the random context. So this differential in the novelty response that's due to the regularity of the sound sequence actually goes away when we suppress the VIP neurons. This does not happen when we suppress either parvalbumin or somatostatin neurons. There is a recovery of neuronal responses, but it's roughly equal for in the regular and the random context. So it seems that the VIP neurons are actually providing this context-specific differential signal to the cortex. VIP neurons indeed actually receive inputs, both neuromodulatory and also inputs from higher cortical areas. So it's very possible that the regularity of sound context might be computed somewhere outside of the primary auditory cortex, and then transmitted back via the VIP neurons. So this is a recent study that we're currently writing up for publication.
Clarification question. So on the previous slide, what was tuning? Is that green part?
Oh, tuning is when we show just the stimulus alone without the context sequence.
Of the excitatory neurons that are tuned for a specific.
Oh, which neurons we select? So these are not specifically excitatory neurons, but they're probably mostly excitatory neurons. Yeah.
Did you expect to get an opposite effect by inactivating SST, since in your diagram they are inhibited by?
Yeah, and we do get the opposite effect. So this, oops, sorry. So when we inactivate PVs or SSTs, the responses increase. But we just don't see this differential effect that's selective to the regular context.
Activation on every trial, or?
No, the inactivation is on the subset of trials. So that's why we're able to compare between, sorry. Okay, that's why we're able to compare the responses on light off and light on conditions.
Yeah, inactivation is only on deviants, or?
It's only on deviants, yeah. So yeah, because otherwise the problem with this is that there's so many different permutations that we can't do it on, we would like to also run it on the standards, but then you have a very long stimulus set, so yeah.
Have you looked into the tuning properties of the novelty responding neurons? So I was wondering whether the neurons that are tuned for specific frequencies are having more either stronger novelty responses compared to untuned neurons. Is there any
So, well, here this is so we select so this is not frequencies, because here it's these frequency modulated ripples, but that's what we do. So we can do that over the whole population, or we can select neuron, only the ripples for which these neurons are tuned, and then look at the novelty responses there. So that's what we usually do. We select the neurons that are actually already responsive to these ripples. Yeah. Yeah.
Are these experiments all from animals who are hearing these sounds for the first time?
So yeah, that's a really good question, because we repeat them, we run them we habituate them, and then we repeat them. So no, yeah,
So
we habituate them. We habituate them to the whole stimulus. Yeah. And then we image from the same animals for several sessions, for several weeks.
Have you ever seen anything
So,
change over this?
We haven't tracked that very carefully because if you want to track it, you really want to track the neurons, and then that's a whole different experiment. So we ran that experiment in a different context with just exposing, and many groups have. So neurons do reduce, like there is a reduction in the responses over days when you play exact same sounds. Yeah. So this, theoretically, this is on the already adapted brain. Yeah.
So the tuning, is that with light? What do you mean exactly? I'm not sure I follow that gray term. What does tuning mean here? Is it with light on, light off?
Oh, this is just baseline responses. So this is if you on this here, this is just responses to the ripples, them individual ripples. Yeah. So that's what we use to calculate the kind of baseline versus novelty response.
And in that plot where you have all the conditions, the tuning is without opto. That's just.
Yeah, and then we don't have opto.
Then you have reversed it, right? I guess it was going negative just because you have reversed, not that it actually goes negative.
Why it's negative? For some reason in the cortex, we see these negative calcium responses for most neurons. We can isolate the population of neurons that have positive calcium responses. You get the same effects. So we kind of could show it in many different ways, but we do keep the negative responses separately from the positive responses. But for these ripples, most of the responses are negative. Yeah. It's consistent with other groups like your Keller, I think. They have very similar
If
responses.
you go to that slide where you had the original, the one that you had.
At the end,
The one-
yeah.
So things here are actually very similar to visual cortex. So
Yeah.
referring to our work at the Allen Institute,
Mhm.
SSC, VIP, but that tuning is very. Yeah, I thought you had it was reversed for the visualization. So I'm.
Yeah, I can't tell you exactly why it's negative. With electrophysiology, we get a much smaller percentage of neurons that show reduction in the firing rate with the same exact. So we did this actually experiment using electrophysiology. We get the same effects, but we don't have, and we get some neurons that are suppressed by these stimuli, but not the same percentage. So, all right. So, so as I mentioned also, a big goal of our lab is to try to integrate our results into computational circuit level models. And so what we try to do is to take our results on the role of parvalbumin and somatostatin neurons in stimulus-specific adaptation and combine them with results from other laboratories. So here we looked at at kind of somewhat different findings from the Hasenstaub lab on the differential function of somatostatin neurons and parvalbumin neurons in something called forward masking, which is another form of auditory adaptation. We also had another study on the differential role of somatostatin neurons on adapting to repeated sounds. And we also tried to integrate this with the study on enhanced functional connectivity with parvalbumin activation from the BAO group by Liberty Hamilton. And so with these, and so what was bothering us at the moment when we did this was that many different laboratories were looking at the function of these inhibitory neurons in auditory processing using different paradigms, and they would find different effects. And so we asked, well, actually, can we use all of these results to try to constrain our understanding of what this microcircuit, of how this microcircuit is wired up. And we put together a model of PV, SST, and excitatory neurons with various types of synapses. So they're connected to each other. There's going to be depressing synapses or facilitating synapses. So we put what we knew from the literature and kind of did the sweep from the models to try to see whether we can constrain those parameters and account for this diversity of the results that we saw in the literature. And indeed, we found that we could recapitulate our results on stimulus, for example, our results on stimulus-specific adaptation. And not only that, we could also make predictions for what's going to happen when we activate these different types of inhibitory neurons. The same for forward masking. We were actually quite happy that we could integrate our results with the results from the Hasenstaub laboratory in a kind of unified circuit model. And we're now working on kind of extending these models to include kind of the diversity of cell types and really put them in the context of behavior. And in order to do that, we have been trying to understand how inhibitory neurons control cortical codes more generally. So I think some of you maybe have heard about this notion that the auditory cortex functions in the sparse firing regime. And unlike the visual cortex, so in the visual cortex, you stick the electrode in and you will see visually evoked responses. So in sensory cortex, even better, you stick your electrode in, you actually know exactly which whisker to activate, you'll see responses. In the auditory cortex, the neurons are tuned in a much more, the auditory responses are much smaller. If you stick the electrode in, you actually sometimes don't get any responses. It's very frustrating. But there's this idea that the auditory cortex really functions in this sparse distributed regime, where different sounds are encoded by small populations of neurons. If you take this representation to a limit, you get to a localist representation. This is something that you can think of as, say, your classic grandmother cell. One cell represents one stimulus, another cell represents another stimulus. On the other end of the kind of coding spectra is a distributed network where overlapping populations of neurons represent different stimuli with different levels of population activity. And we can think of these representations as neuronal spaces such that in a localist representation, if we take a high-dimensional space where each dot represents the activity of all of the neurons that we're looking at in response to a certain stimulus, in the localist representation, you're going to have an orthogonal representation of different sounds, where basically non-overlapping populations of neurons are going to be represented different signals, different stimuli, whereas in the distributed representation, you will have very much aligned responses to different stimuli, just with different magnitudes. We were very interested in this, and we hypothesized that maybe the function of inhibitory neurons is to shift the population dynamics between these different regimes. Of course, these different coding schemes can be useful for different tasks. For example, if you really want to discriminate between different things, you want to be in the localized regime. But if you want to be able to generalize, you want to be in the distributed regime. And so what we wanted to see was whether if we image the activity of neurons in a network in response to different stimuli and activate different subpopulations of inhibitory neurons, how would this affect these different codes? our hypothesis was that activating somatostatin neurons should actually make the populations more distinct, so drive the network into a more localist regime. So these, if we think about these population vectors, we expect a larger angle between them, whereas activating the VIP neurons would actually drive the network into a more distributed coding regime, where overlapping populations will be more, this two stimuli will activate overlapping subpopulations of neurons. And so we repeated the experiments I showed you before, which is 2-photon imaging of cortical activity. But on a subset of trials, we would activate either somatostatin or VIP neurons. And as the probe stimulus, using optogenetics, and as a probe stimulus, we used noise of different sound pressure amplitudes. There were two populations of neurons: monotonic neurons, which increase their activity depending on how loud the noise was for different noise bursts, and non-monotonic neurons, which had the preferred sound pressure level. And using these predictions for whether the population responses should be more localist or distributed, we could actually map the expected changes to the tuning curves of individual neurons. And in fact, we found that the changes that we observed really supported these predictions. And we also found that if we measured the angles between the representations of noise at different sound pressure levels, we also saw that, in fact, activating somatostatin neurons increased these angles between the responses, whereas activating VIPs decreased this population angle. So similar populations of neurons that were activated by the stimulus became more similar, but the amplitude differences became amplified. So in a way, this suggested to us that these neurons are actually acting at a population level, shifting these coding dynamics between localist and distributed representations. Yeah.
So with the SSC kind of makes sense because it's inhibited, so it's making it.
Yes.
With the VIP, there is the disinhibition. But then I'm curious why it makes it even more general. Do we have like some lateral connections here that it makes excitatory neurons closer to each other? So yeah, I'm curious about the VIP and why.
I don't know why it happens, so this was very much like a study. We're just looking at the network, and this is what happens. You can imagine different types of connection schemes. So VIPs at the basic level, right, they suppress somatostatin neurons, but they also suppress parvalbumin neurons. So if you remove that inhibition, you actually get both increased activation of excited neurons and also a broadening in that activation. So, but we didn't really map that would be kind of the next level of study, would be to say, well, how do the subpopulations of somatostatin neurons and parvalbumin neurons change when we activate VIPs? And can we then map it onto excitatory neurons? We started doing that. We started kind of trying to integrate more spatially, how far out do these effects go? Because with 2-photon, we have the spatial data, but we didn't really get it to a conclusion.
Sometimes
We're activating them, yes.
you try with PV as well. I'm curious.
We didn't actually do PVs on this study because I don't think it would have done much, because it simply down regulates the activity of excitatory neurons. So, great. So yeah, so I showed you, hopefully I convinced you that cortical inhibition plays a fundamental role in auditory computation. controls frequency discrimination acuity, adaptation to various forms of statistical regularities and novelty responses. And I hope that we can now really push these studies to understand this the excitatory inhibitory dynamics at a population level in the context of various behaviors. And so I want to acknowledge the people who did this work over the years. So the work on stimulus-specific adaptation was done by Ryan Nathan and John Briguglio, who were graduate students in my lab. The cortical model was done by Youngmin Park. Shamao did the work, or is now currently doing the work on adaptation to regular and random context. And Melanie Toban and Janki Chef worked on the population dynamics study. So thank you. I also want to take this opportunity to advertise the EARS seminars. So there are some EARS devotees in the audience here, but this is an online seminar series that's run out of my lab, and we meet on Tuesdays, so you guys can come online. And we have a trainee session coming up where we solicit abstracts from trainees and then select 4 abstracts for short talks. And then we'll have seminars by Jenny Groh, Pierre Apostolidis, and Maria Chait. And also I co-direct the Cahal course on computational neuroscience. We don't have it this summer, but we'll have it next summer. It's a 3-week summer course in computational neuroscience at the Champollion Center in Lisbon. It's really fun. So those of you still in the training stages should apply.
Maria, thanks for that. This might be asking again the same question Ming asked earlier about, do you think the results here would be different if instead of inactivation, you were able, because fundamentally in that case, you're changing the spontaneous activity, but if you were instead hold those neurons fixed at their spontaneous rates, do you think that that would still produce the same kind of observations you're having?
Well, I think so, yeah, because we always look at the responses, both the actual responses and also subtracting the spontaneous activity. So I don't really.
The spontaneous rate must have some effect on the overall sort of tone or dynamics of the circuit. So by inactivating them, you're maybe changing that balance.
That's true, yeah. Yeah, I don't really know how to disentangle them. So it's kind of maybe a form of that question could be, well, are the inhibitory neurons acting on the inputs to the, like, are they acting on the actual transformation into the firing rates? At what stage of integration of information are they acting, right? If they're just making the excitatory neurons more responsive, so they're changing the response gain, or are they offsetting the activity level, so they're effectively lowering the threshold for the responses, or are they amplifying the inputs? And I guess it's all of the above, yeah. So, right, yeah, but I like that question. We think about that a lot, yeah. Yeah.
Mine
Uh-huh.
from Abigail McElroy said all of these studies seem to be on untrained mice and the natural responses of the brain to sound. Have you looked at whether or not affecting local circuit dynamics in this way changes behavioral output? What can you get the mouse to report on its perception?
Yeah, we have. So we have really been pushing kind of the limits, I think, for what we can train mice to detect, to report, and to detect. In one set of studies, we have trained mice to detect a sound at different time points after transition from high to low contrast noise. And this is a study that's been recently published, where we find that adaptation, cortical adaptation actually maps perfectly onto the mouse perception. So I was very proud of that work because it connects efficient coding in the brain to mouse behavior. And we're now investigating the role of inhibitory neurons in that process. So we're actually really trying with that study, we can really map whether there is the shift in the gain in the cortical neurons driven by inhibition and how that affects perception. In another set of studies, we've training mice to detect sounds in regular and random contexts. And we can show that indeed, they are detecting these novel sounds, but it is really hard to do those studies. So we have been trying for a few years and we have some interesting results on that, but it's not really ready for publication yet.
I was curious about the frequency tuning
Mm-hmm.
and exactly when in development frequency tuning emerges in auditory cortex and how that aligns with the maturation of inhibition.
That's actually something that has not been studied. And so we're very interested in studying that. So we are actually starting a line of work trying to understand the role of excitation and inhibition in autism spectrum disorders. So in autism spectrum disorders, there is changes in hyper or hypersensitivity to sounds. And also, there's also differences in inhibitory excitatory balance. And so we're going to try to look at different developmental time points and try to map that exactly. Yeah, that's going to be super interesting. Yeah.
About your local versus distributed code, isn't there a map of frequency space in artificial? And I'm
Sure.
wondering if you disinhibit the circuit, I would expect that localized region to just get bigger as opposed to the code getting.
So that's a really good question. So we can gather that with these kind of more precise stimuli and more precise manipulations. In the study that I showed you, we just played white noise. So it should activate all of the neurons, kind of to the extent that they are responding to that. But that's a really good question. Yeah.
So it's past 12:15 now, so let's stop
Okay.
there. Yeah, thank you so
Okay,
much.
super. Welcome.
I was like, am I gonna